back for a relatively short video on innate immunity. So we're going to start by listing sort of the key components of innate immunity. And again, the difference between innate versus acquired, also known as specific immunity, is that the innate immune system components are ready and active immediately upon infection. The acquired system requires a sort of a ramp-up period where those cells recognize the infection and then are duplicated, cloned, and activated to produce an army of cells, very specific, and again, cells and or antibodies, which are very specific to the invading microbe. All right, but so back to innate immunity. Those are the components that are ready to go to fight infection right from the beginning. Right, and the first one's fairly simplistic. That's the epithelial barriers. These are the membranes that line the open spaces of our organ systems that are continuous with the outside world, i.e., they are prone to infection because of that exposure to the outside world. Skin is an obvious one, but the skin only has you have about 2 square meters of surface area of skin. Whereas if you add together the surface area of your lungs, your GI tract, and your urogenital tracts, you have about 100 square meters. So dramatically more surface area that needs to be protected sort of in the inside compartments of the body that are yet exposed to the outside world through these open passages. So those epithelial barriers are important because that is the wall that has to be breached by a microbe to invade and infect the human body. Now, that's not to say that we don't fight microbes that are on the outside of that epithelial barrier. We do. Many of those epithelial cells release antimicrobial peptides, which tend to inhibit microbe cell division and proliferation. In addition, in some places, especially in the GI tract, there are secretions that aid digestion, but also tend to kill the microbes. An easy example there is the acid secretion by the stomach. It tends to kill a lot of the microbes that we ingest with our food. Okay, so epithelial barriers, pretty straightforward. Let's go to the professional phagocytes, right? There are a few other cells that will phagocytose things, but the 2 main ones that are really pretty good at it are the neutrophils and the macrophages. Right, so the phagocytes include the neutrophils and the macrophages. Now, they are a little bit different. They both phagocytose foreign entities, whether it be free-floating foreign antigens or things like whole bacteria or even virally infected cells. They're a little bit different, though, and I mentioned this in class, in that the neutrophils tend to circulate in the bloodstream, and then upon the chemical signals that get released, coming across the chemical signals that get released, they will what's called extravasate, extravasation. Let me write that down. And they both, these are the macrophages are derived from monocytes. Right, that's their precursor. And both of them have to undergo what's called extravasation. I mentioned this in class. I may not have used this particular word, but that's the word that means the cells have to cross we actually do it in venules, small veins, but cross the blood vessel wall to enter the interstitial space where the infections generally are occurring, right? The interstitial space for instance, underlying the lining of your digestive tract, or the lining of your respiratory tract, or the lining of your urogenital tract, or the lining, or your skin. Anyway, so the neutrophils largely will be drawn out of the bloodstream in response to the chemical signals that occur, and again, coming from things like basophils or mast cells that recognize the infection and initiate inflammation. But those chemical signals draw the neutrophils out of the bloodstream to where they can fight, i.e. phagocytose, the invading microbe. Lots and lots of them, right? But they're a little slower because they have to sense the infection and then be drawn out of the circulation before they can actually act. In contrast, monocytes will extravasate sort of spontaneously without any real stimulus to do so, and then just sort of hang out in the tissue, inactive, as tissue macrophages. There are many fewer of those relative to the neutrophils. However, there they are. They're sitting there in that tissue, sort of where the infection is likely to occur. So they're ready to go as soon as the invading microbe sort of crosses the epithelial barrier. Boom, there they are, and they're phagocytosing it. The neutrophils will show up a little bit later and then help them, because the numbers in the macrophages might not be enough to eliminate the infection. Okay. The one other thing we have to mention about phagocytes is they are what are called antigen presenting cells, or APCs. Right? So the antigen presenting cells, what that means is that they not only phagocytose these foreign bodies and kill them, and get rid of them, but they also sort of digest them, take samples of them, and then show them on the surface of their membrane using MHC proteins. And they use what's called MHC class 2, an MHC2 protein to do that. So what they're doing is they're taking these antigens, in this case foreign antigens, from the microbes that they're eating, and they're showing them on the surface of the cell. Well, who are they showing them to? Largely, they're showing them to the naive B and T cells, linking the innate and the acquired immune systems, right? So they're taking what they just ate, using the MHC, remember I called it a flagpole, right, to show the flag, the foreign antigen in this case, because they phagocytose something foreign, show it on the surface of the cell where the naive B and T cells, which are usually hanging out in the lymph nodes after they've been matured in the bone, and for T cells in the thymus. But those cells sort of hang out in the lymph nodes and sort of wait for something to show up, right? Well, those foreign antigens show up oftentimes on the surface of these antigen-presenting cells, whose job it is to sort of, move from the interstitium into the lymph nodes and sort of say, hey, anybody have a match for this, right? I just gobbled up this thing, right? And then the matching B and T cell groups that have a receptor that binds that foreign antigen will respond and become activated, and then we've got an acquired immune system response that we'll have to talk about a little bit later. Okay? So those are our professional phagocytes and their job as antigen-presenting cells. Okay, let's do another one. I'm going to run out of room. I'll have to just keep going here. I'll continue with my list. That was number 2. Epithelium was 1. Phagocytes is 2. 3, let's do complement. Right? Things that are complementary to one another sort of work together and probably do very similar things. So what is complement, or the complement proteins, complementary to? They're complementary to antibodies, right? So they sort of do what antibodies do in certain ways, okay? Now, so what is complement? Complement, remember, is not a cell. Complement is a series of proteins. It's actually a series of enzymes, largely. And you get this sort of cascading effect once they're activated following chemical signaling again, the similar signals and markers that show up with virally infected cells, or recognition of bacteria and things like that by the phagocytes, and so the complements become activated. And what they do essentially is two main things really. They opsonize, right? And so remember I used the term, or the synonym basically, label, right? So complement proteins upon coming across something that's been identified as foreign, will bind to it and label it. And again, they're labeling it for the phagocytes to come and attack it. In addition to, and actually this opsonization will aid the antibodies sticking to it as well, right? So antibodies that would come across something that's opsonized would bind to it. But so they become opsonized, but we also form the MAC, right? The membrane attack complex. And what this is it's a series of proteins that once activated, again, complement is more than 20 proteins, and a group of them will become activated and insert in the membrane of this virally infected or foreign cell, like bacterium, and form a pore. And that pore allows the inside and the outside of the cell to, the gradients to diffuse across the membrane, dissipating potential energy of the sodium gradient or the potassium gradient, or electrical gradients, right, that are essential to the survival of that cell long term, right? So the membrane attack complex tends to kill the cell outright, as opposed to opsonizing, which is labeling it for somebody else to kill it. Okay, that's complement protein. Let's talk a little bit about interferon. Interferon is released by several different cell types upon activation that follows infection. And there's one very specific cell type that releases it. It's a special type of what's called a dendritic cell. Dendritic cells are another version of the antigen-presenting cells. They're phagocytic too. Those cells, that specific dendritic cell makes lots of interferon in response to an infection, and interferon does a couple of key things. Right? Interferon interferes with protein production. Right? And so basically what it's doing is it's the cell survival depends on, so think about a virally infected cell. That virally infected cell is making viral proteins, right, out of its own control. The virus has sort of like hijacked the cellular machinery to make its own proteins. But it also is still making its own proteins. And so what the interferon will do is interfere with its ability to produce proteins in general. And so it will stop making viral proteins, but it also stops making its own proteins. So that cell kind of gets shut down. It can't replicate the virus anymore, but it also can't really survive for very long because it's not able to recycle and go through protein synthesis on its own. In addition, interferon initiates apoptosis in these virally infected or cancerous cells, and so we end up sort of shutting it down in terms of its protein production, but then also telling it basically to commit suicide, after which, once that happens, the phagocytic cells will gobble it up as a dead or dying cell and remove it, okay, including its internal viral particles that it's been producing. Okay, that's interferon. Last one I think we'll hit is the NK cells. Remember, natural killer. And they're called natural killer cells because they don't actually require antigen presentation the way that the B and T cells do to become active. In other words, it's naturally a killer cell. It doesn't need even to be really stimulated by anything. It needs to be stimulated by the fact that it recognizes those microbe associated molecular patterns, right? The things that make a microbe look like a microbe instead of like a human cell. And so once these NK cells recognize that, and through other chemical signaling from other activated innate immune system cells, the NK cells will secrete two things: perforin and granzymes. Perforin acts a lot like the membrane attack complex. The perforin proteins perforate, form a pore in the membrane of this infected, damaged cell, and dissipate the transmembrane gradients, right, potentially killing the cell. This is sped up a bit by the granzymes, which are basically enzymes that come from granules, thus the name granzymes. And those enzymes initiate and activate apoptotic genes and initiate apoptosis in those infected or abnormal cells, again causing the cell to initiate self-destruction, after which it'll be gobbled up by a macrophage. Okay. The only thing I didn't say a lot about here was inflammation. Remember, inflammation is sort of a key to initiating, attracting, and activating a lot of these innate system elements, right? Mediated by things released by damaged cells, as well as the output of mast cells and the arriving basophils that'll respond to that, right? And so inflammation causes the redness and the swelling and the pain that we get associated with like things like a mosquito bite or a bee sting. And a lot of that is initiated by the eicosanoids, right? Remember the eicosanoids? They're one of our chemical messengers. And so one of those molecules initiates pain, another initiates vasodilation. One of them causes the histamine, right? Is one of the key components released, causes the blood vessels to become more permeable, which allows the white blood cells to more easily move extravasate. That was on the last board, right? Extravasate. The endothelium becomes a little bit more permeable. The white blood cells are able to more readily move out of the vessels and into that inflamed site to attack the infection. Okay.